today I want to tell you about some of the work that our lab has been doing on the development of amygdala-prefrontal circuitry. We spend a lot of time thinking about not only development, but the function of development. So what is going on during periods of immaturity that have long-term implications for adult functioning? And we spend, today I will be focusing very much on the connections between amygdala and medial prefrontal cortex, because this circuitry forms the foundation of emotion regulation behaviors that we see in maturity. Now, the amygdala is this deep structure in the brain that is involved in emotional tension and supporting emotional learning, and in maturity has very strong bidirectional connections with medial prefrontal cortex that by and large facilitate coordination of those amygdala inputs, often for the purposes of regulation of those amygdala signals. Those connections, or that bridge, takes a long time to develop in the human, as any parent will testify to. So we're not only looking at this during development, we want to look at this system as it's cooking, so to speak. And we're doing that because we know that there's this very strong association between early experiences, both positive and negative experiences, and emotional functioning in adulthood. And because of the prolonged plasticity of amygdala and prefrontal cortex, that may serve as a very promising candidate for understanding a potential mediator of those links between early experiences and later emotional functioning. And we want to do this while the system is developing, because there are many mental illnesses that are associated with difficulty in emotion regulation, and they tend to show their peak age of onset after the end of childhood. So we want to look in the antecedent period while these systems are developing to understand where these individual differences come from in adolescence. So today I'll be talking about normative changes occurring during development in this circuitry, thinking about the role of caregiving, and finally turning to early caregiving adversity during development and how that affects trajectories of this system. So I'll start with an earlier paper from our lab where Dylan G., who was a graduate student in my lab, started by using a very basic tool in fMRI, which is to present fearful faces to participants. And she invited participants typically developing typical caregiving history between the ages of 4 years old and 22. And she presented these fear faces and then performed standard pre-processing on everyone's brains and took all the brains between 4 and 22 years old and smushed them together and looked for age-related changes in reactivity to these fear faces. And she observed, this was a voxel-wide approach, which means there were no a priori restrictions here. She saw that there was this negative correlation with age in the amygdala, in the right amygdala. And so I'm showing you those data here. This is age on the x-axis, and on the y-axis is amygdala reactivity, so the change in bold signal in response to these fear faces. This builds nicely on the work of people like Amanda Geyer, who showed that during development we have these strong amygdala signals, and we've just taken it back further. So the thing to notice here is that the younger we go, and the youngest we went here was 4 years old, the stronger the amygdala signal is to these fear faces. Now, fear faces are great because they're a relatively innocuous way to target the circuitry in children. It's hard to study fear systems in children and have them keep coming back to your laboratory and have their parents sign the consent forms. But we've also, there are drawbacks to using fear faces that I'm happy to talk about. So we've also used fear conditioning paradigms where we pair a geometric shape with something aversive, and we still see this really strong amygdala response in 4-year-olds. Now, we are interested in not only the amygdala, but how the amygdala starts to connect functionally to medial prefrontal cortex. And we can assess this in using fMRI data with functional connectivity. So for those of you that are less familiar with this method, we can pull the activity or the time course in one region, like the amygdala here in green, and the medial prefrontal cortex in blue, and assess the degree to which they're correlated with each other. These are correlated time courses, so we would say that they're functionally coupled with each other. So when we can collect this type of functional connectivity data at rest, which is to say when subjects are doing nothing. We just have them lie in the scanner for 6 minutes and collect these time courses. And we did this again with subjects ranging in age from 4 to young adulthood. And this is the work of Laurel Gabbard-Durham. She places a seed in the amygdala and then looks for age-related changes in functional connectivity. Now, we saw what many others have seen in typical adults, this strong connectivity between amygdala and medial prefrontal cortex. But in children, we didn't see that connectivity, and that doesn't start to emerge until after childhood. Now, that positive connectivity that you see between amygdala and medial prefrontal cortex is what is typically observed across many studies when connectivity is measured at rest. But if you can measure connectivity when people are engaged in some task as well. So for example, in response to those fear faces, what does connectivity look like? In adulthood, what people typically find is you get strong connectivity, but it's now negatively correlated. The amygdala and prefrontal cortex are negatively correlated with each other. So the suggestion is that when prefrontal cortex increases its activity, amygdala decreases its activity and vice versa. So in adulthood, you see positive connectivity at rest and negative connectivity or negative correlations at rest, in response to a task. So in fact, the more negative that connectivity in adulthood, the more the amygdala quiets down over the course of the testing session. So this is suggestive of a regulatory type of relationship between amygdala and prefrontal cortex, and that's been substantiated by animal studies and lesion studies. Not only is the amygdala quieting down more, these individuals tend to have lower trait anxiety, lower autonomic arousal, and so on. So the healthy, robust, mature pattern we would expect to see is this negative correlation, negative communication between amygdala and prefrontal cortex. So when we look across this wide developmental range, we again place a seed in the amygdala and then look for connectivity changes with age in response to those fear faces. And we see the medial prefrontal cortex showing up as being a significant area associated with age. And these are what the data look like here. So in adults, we find what many people find, this negative correlation between amygdala and medial prefrontal cortex, suggestive of a regulatory relationship. In older and younger adolescents, we are still seeing that negative correlation, although you can see it's less robust than it is in adults. And that's consistent with the poor emotion regulation that adolescents have relative to adults. But when we look in children under the age of 10, we don't see that negative correlation. We're seeing a positive correlation in response to stimuli. So in other words, the nature of communication between amygdala and medial prefrontal cortex is different in childhood than it is thereafter. Okay? So we've since become very interested in this developmental valence switch in the way that amygdala and prefrontal cortex are talking to each other. So in other words, if in the adult state we're getting this negative correlation, prefrontal cortex is increasing its activity while amygdala decreases and vice versa, the implication is the child state has both of them moving in the same direction. So the communication is different. So why would we, what interpretation can we draw from these types of data? So I just wanted to share with you the way that we're currently thinking about what these data could mean. So given the hierarchical nature of brain development and the fact that the amygdala develops earlier than cortical regions, it may be that during early childhood, the connectivity is reflecting very much communication from amygdala to prefrontal cortex. And so it's as if early on the amygdala is sort of reaching up and knocking on the prefrontal cortex saying, I'm here, this is who you're going to be living with for the rest of your life, right? So that over time, you can start to instantiate these more top-down regulatory influences that we see in adulthood. While we can't conclude that from our data for sure, there are tracing studies, for example, from rodents showing that these amygdala-originating inputs to prefrontal cortex are emerging developmentally earlier than prefrontal cortex returning back to amygdala, suggesting there might be merit to this idea. So putting things together, if this is age on the x-axis here in years, we're already seeing in childhood this really robust amygdala response. But that robust amygdala activity is happening in the absence of adult-like connections with medial prefrontal cortex, which we don't see typically emerging until 10 or 11 or so, which makes childhood a very interesting time for us to think about because the amygdala-prefrontal phenotype seems so distinct from later ages. Now, we are very interested in the idea that this circuitry is not developing in a test tube. It's not developing independently. It's developing in a very special environment, which is the caregiver. So caregivers are a species-expected stimulus for the human. And I'm thinking about the work of people like Myron Hofer, who showed in rodents very beautifully that different aspects of the mother had unique and specific effects on regulatory influences on the physiology of the pup. So we can extend that idea further and think about the parent as providing a regulatory influence over the central nervous system. So certainly in humans, caregivers don't just have to be moms, they can be dads, grandparents, and so on. And this is not unique to humans. Many animals spend time with their caregivers. But what is potentially unique about humans is how long we spend around our caregivers, right? This is a really bizarre design by Mother Nature in many respects because it's a huge energy suck to raise another human being, right? I don't know what the number is, but someone computed how many calories it takes to raise another human being, and it's enormous. So there's got to be some big payoff to this large amount of time that we spend around our caregivers. And we're motivated by theories that argue that this caregiver presence helps keep us regulated during a period of central nervous system immaturity. And so we can think about the caregiver as scaffolding our central nervous system, providing that regulation input while our regulatory systems are still developing. So again, we turn to the rodent research, which has really provided some compelling data showing just how much how much influence the parent can have over these emotional systems. So this is summarizing the work of Regina Sullivan, who's at NYU. And if you're not familiar with these data, in brief, the functioning of amygdala right after birth is pretty quiet and very different than the functioning of the amygdala during adolescence and adulthood when it's reactive. However, there's this period in between, a transition period, where whether the amygdala functions like the newborns or more like the adolescent and adults is dependent on the mother being in the nest or not. So when the mother is in the nest, amygdala will act like the newborns. When the mother is out of the nest, amygdala will act like the older animals. So the mother's presence is buffering against amygdala reactivity during development. So we have this period of a mother-dependent or parent-dependent sensitive period. And the way that this plays out behaviorally works like this. You can administer a threat conditioning paradigm where a peppermint odor, for example, which means nothing to the pups initially, is paired with a mild foot shock. And then you place the peppermint odor at one arm of a Y maze and a control substance on the other. Now, in the rat pup, if they're conditioned without the mother present, they will do what you and I would do. They would avoid the peppermint odor and go in the other direction, and that's what's being shown here. They're not really choosing the arm with the peppermint odor. However, if you do the same conditioning with the mother present, and this is, notice, an anesthetized mother, so it's just her physical presence. Her presence should keep amygdala down, and amygdala is the neural substrate of aversion learning. So if she's keeping amygdala down, then her presence should be buffering against fear learning. And in fact, that's what's observed. So the animal is now counterintuitively approaching the peppermint odor. So not only are they not avoiding it, but they're approaching the peppermint odor. So there is a really elegant set of studies showing that this is a good reminder that the developing organism is not just a miniature adult, but they have different developmental goals. The goal of a young pup is to form a preference to its surroundings. So even in the context of something like a mild foot shock, if the mother is present, amygdala remains quiet, leaving open competitive learning systems to take over learning, and so you get this seemingly bizarre but developmentally appropriate preference learning. So this whole process is mediated by CORT, the stress hormone CORT. In rodents, mother's presence is keeping the stress hormone CORT low, which is preventing amygdala engagement during fear learning, and so you get this preference learning during development. Now, given that humans take so much longer to develop and we hang around our parents for so much longer, might we expect to see some evidence of this parental buffering even in childhood? So Cam Hostenar, who is now here, when she was in Megan Gunner's lab, asked this question using cortisol as her outcome measure. She had children come into the laboratory and undergo a Trier social stress test, which, if you're not familiar with it, is asking your participants to give a public speech, which is a terrific way to elevate stress hormones. And children did this either, they prepared for this talk either sitting next to a stranger or sitting next to their parent. If they sat next to a stranger, they showed a typical CORT response, that blue line. But if they sat next to their parent, that response was buffered. Now, importantly, this buffering was not observed in adolescents. Adolescents showed that typical mounting of a CORT response regardless of who they were sitting next to. We wanted to extend this idea and look at what's happening at the level of the amygdala. So recall that I started off earlier showing that we get this really robust amygdala signal in early childhood. This is to fear faces, and this is to that fear learning paradigm I was referencing earlier. But these data are collected when children are not with their parents. So might we expect to see something very different if we give children access to a parental stimulus? So we showed children pictures of their parent and contrasted amygdala signal when they were looking at somebody else's parent. Now, during childhood, we saw that presentation of the parental stimulus was decreasing amygdala reactivity relative to somebody else's parent. So we're interpreting this currently as evidence of parental buffering of amygdala signal. Adolescents did not show this buffering effect. Adolescents' amygdala was not changing as a function of whether they were looking at their parent or somebody else's parent. That's not to say that parents aren't important during adolescence, but it may just be at the level of the amygdala, parents are less effective in changing circuitry. We also found that presentation of parent was increasing the strength of the negative correlation between amygdala and medial prefrontal cortex. And that effect was moderated by attachment security. So it might be that when parents are unavailable, which happens routinely, parents go to work, children go to school, but they always come back. When parents are unavailable, amygdala reactivity may be normatively high, and when they become available again, amygdala reactivity goes down. So we're using these types of data to try and build a testable working model of the interdependence between this emotion regulation system and the social environment. So what we're thinking right now is, at least during childhood, we haven't gone younger than 4, so this might extend backwards, there's this time when parents' presence is having this highly modulating effect on amygdala reactivity and associated fear behaviors. And I have up and down green arrows here because my intention is not to imply that parents should be around all the time to block fear learning. In fact, parents should go away, right, and then come back so that you have that elasticity throughout the day. But that elasticity may actually serve an important function of toning the system, in training the emotion regulation system, so that as the system, the plasticity of the system is lost as we get older, the nature of functioning out in adulthood may reflect the history of that modulation during development. And so if this is indeed a sensitive period, we have some ideas about possible mechanisms underlying that sensitive period. So for example, when parents are unavailable, we might get increases in cort and amygdala and associated fear. And when parents become available again, we will see decreases in cort, increased coupling between PFC and amygdala, and decreased fear. So what's the agent of change here? We have age on the x-axis here, but that doesn't tell us much about what the agent of change is. And so there are lots of candidates. We get asked a lot about puberty as being an important factor, and certainly that's an important one. Another potential agent of change is the increasing independence that we have from caregivers. So there's a huge shift across this time in terms of how much time we're around our parents. So when my kids were infants or toddlers, I felt like I had human beings stuck to me all the time. And then one day they were both on the floor and I could read a newspaper. And now they're off in their room, and then soon they'll be out the door, and so on. So that could be a powerful agent of change. So to address this question in a more extreme group, we've had the privilege and the opportunity to work with families that have adopted children who had a very atypical early experience in terms of separation from a caregiver. So we've been working with families that have adopted children from institutional care abroad, and then the families are now living in the United States. So before I go any further, I would like to point out again and again throughout this talk that there's tremendous heterogeneity in outcome. And I will try in the data to point out how we're starting to think about this heterogeneity. Children who experience this type of institutional care, even in the best of circumstances, are experiencing a suboptimal caregiving situation for humans because the caregivers are staff members who rotate shifts and go home on the weekends or get fired, and the caregiver-to-child ratio is very poor. So many children upon adoption exhibit a number of developmental delays. However, the families that adopt internationally not only tend to be a well-resourced group of families, but more importantly, have a great desire to provide care, and because it's not easy to adopt internationally. And so it's an ideal situation to see tremendous growth, and indeed there is tremendous catch-up in many domains following adoption. However, one of the areas where children may continue to struggle is in the domain of emotion regulation. So we're viewing this as a potent stressor for the human infant. And if we think about it as a stressor, then we can turn to the stress literature, which can give us some important insights and generate hypotheses. So these are data from a different species, different stressor, et cetera. These are adult rats using a chronic immobilization stressor, but there are important take-homes here. So following administration of stress, amygdala shows a greater number of spines, and amygdala will show this hypertrophy, this larger cell size of the amygdala, which is accompanied by greater reactivity, which is very different than what's happening in neighboring hippocampus, which tends to show decreased dendritic complexity, smaller cells, and less functional cells. So I think this is an important contrast because it suggests to us that stress doesn't have a uniform effect on the brain, but rather what stress is doing is it's informing the brain or instructing the brain, sculpting the brain to be more vigilant for future threats out in the environment. If we look during development, we see more of that idea. It's even more pronounced. So I'm about to show you a number of panels up here, but the punchline is the same across a number of labs. Now, these are all rodent data. Early stress, so in this case it's always going to be separation from the mother, is associated with accelerated development of fear-related neurobiology. So that was a new way for us to think about the effects of stress. Usually stress is associated with the idea of impairment, but instead these data are suggesting if we look during development, we're actually seeing an enhancement of function in these survival-related systems. And all of these effects, again, are mediated by the stress hormone cort. So premature separation from the mother in rodents is associated with premature myelination of amygdala neurons, premature amygdala reactivity, premature retention of fear memories. So typically raised rodents will learn fear, but they have amnesia. They forget it within 10 days. But following maternal separation, animals hold on to that fear memory very well, just like adults do. And there's premature use of adult-like fear regulation strategies, suggesting that medial PFC is starting to develop earlier in these animals. So why would we have this type of developmental phenotype following early adversity? Current thinking about this is suggesting that these differences in the brain might be better understood as ontogenetic adaptations or developmental adaptations in response to these early cues of stress. So it's not that the entire brain is developing faster, but rather there might be neural reprioritization. So when the developing animal is receiving cues that the safe caregiver is not always going to be available, then it may benefit the individual to reprioritize through an activity-based process these systems that are involved in emotion and emotion regulation. So if we look at these differences through the lens of adaptation, then we can maybe understand behaviors better through the lens of trade-offs between benefits and costs. So for example, if these are adaptations, these may benefit the individual in the short term. But if they're benefiting the individual in the short term, is there a potential cost that might be observed in the long run, or a cost that might be observed in other domains of functioning? So we've been asking some of these questions in children who experience this early institutional care. And I also want to point out that in terms of timing, this is an interesting group to consider because the early adversity that children experience is very different than the environment they're living in at the time of testing. So they're no longer living in those adverse conditions. So I mentioned there's significant rebound upon adoption, tremendous heterogeneity, but there's a significant elevated risk for emotion dysregulation, in particular anxiety. So I have data from two separate samples that I'll be presenting today, one from New York and another from LA. And there's a lot of text on here, but what I want to point out is in both, the majority of children were placed in institutional care within the first year, mostly at birth, and adopted within the first 2 or 3 years. So I'll be using the abbreviation PI. This is referring to the previously institutionalized group. And in both the New York and the LA sample, the PI group in black is showing increased internalizing problems and anxiety across a number of subscales, replicating what many groups have. And when we look in the amygdala in response to fear faces, we were able to replicate this amygdala hyperactivity to fear faces, and it seems pretty specific to the amygdala, consistent with the amygdala's early development. Now we know that amygdala is intimately tied with behaviors like eye contact in primates, and we were able to measure eye contact through two means. One was to use eye tracking, which gives us a high precision measure of ocular motor behavior. And we also measured, to increase ecological validity, we measured eye contact during a live dyadic interaction with the parent. And what we observed by both measures is the post-institutionalized children were showing decreased eye contact relative to the comparison group. But importantly, we were also able to see that this was associated with amygdala signal to those fear faces in the scanner. The more amygdala reactivity children exhibited, the less eye contact they were making, and that was true by both measures. So we think that this might be an adaptation children are exhibiting, perhaps to avert gaze, to decrease some of the over-arousal that can be instantiated by face-to-face contact. So to further this idea that what we're seeing may be types of adaptations children are making, we used the BART task. If you're not familiar with this, it's the balloon analog risk-taking test, but we adapted it. And we used this task because we thought it would give us an opportunity to look at different ways that different children approach decision-making in the world. So in this task, you're asked to pump up a virtual balloon on a computer by pressing a button. And every time you press a button, the balloon gets bigger and you earn more money. Now you can keep pumping up this balloon, or you can choose to cash in. If you keep on pumping up the balloon, you'll keep making more money, but it could pop at some point. And if it pops before you cash in, you lose all of your money. We adapted this so that there were two conditions. One was a pink balloon. It was a generous condition. It was a generous balloon. It lets you keep on pumping up and earning more and more money. And the blue balloon was the harsh condition. It would pop right away. This is, subjects don't know this when they start. And so our question is, are there differences as a function of early caregiving experiences in exploration versus exploitation strategies? So exploration strategies would be those where you keep on pumping, you're risking even the balloon popping because you want to see how far you can go. And exploitation would be you're going to cash in even if it means you're not earning as much money. What we observed is the comparison, the typically raised children were exhibiting much more exploration type strategies, whereas the PI group was more likely to exhibit exploitation strategies. However, there were no group differences in total earnings by the end. So they both got to the same point through different routes. And importantly, it was the anxiety itself that was mediating this association. So the association between early caregiving group and how much you just cashed in was mediated by your trait anxiety. Now, if we look at exploitation strategy and total earnings in that generous balloon condition, having an exploitation strategy doesn't benefit you. However, in the harsh balloon condition, where it popped quickly, having an exploitation strategy was advantageous. And here we have the post-institutionalized children in the colored circles outperforming the comparison group, giving us another hint that these differences in behavior as a function of early experiences may play out differently in different contexts. To further that idea of adaptation, we pursued this acceleration hypothesis in amygdala-prefrontal development. So again, using those fear faces, we presented these to children and adolescents in the typically raised group and in the post-institutionalized group, and we observed important age by caregiving interactions in prefrontal cortex connectivity with amygdala. So these are the data I showed you earlier where the typically raised group was showing a more childlike positive correlation that switched for the adolescents to more negative. We were able to replicate that in an independent sample. But in the post-institutionalized children, we didn't see that childlike pattern, but instead saw a more adult-like pattern. They look more like the older adolescents, even though they're between the ages of 5 and 9 years old. We're also thinking this is an adaptation. Even though they look different from their peers, we think it's an adaptation. So I'll explain shortly. So that association between early caregiving and the nature of your connectivity was also mediated by CORT. What we think is happening is this circuitry, the connectivity growing up in the context of amygdala hyperactivity may be recruiting these connections with prefrontal cortex at an earlier age, and if so, we may see the earlier onset of these connections and potential abbreviation of that sensitive period in childhood. So here's where the adaptation piece comes in. Not all of the PI children showed this adaptation, but 61% did. So we looked at those individual differences in relation to anxiety behaviors. Although at the group level, all of the PI children showed increased anxiety relative to the comparison group, if we look at those who showed the adult-like connectivity patterns, they had lower anxiety relative to their peers who didn't show the adaptation. So this is suggesting that the circuitry is doing what we would expect it to do. It's associated with lower anxiety. It's not coming all the way down, we think, because there's still a lot of bottom-up reactivity with which the system has to contend. But this is suggesting that, at least with current anxiety, having this adaptation is helpful. So putting this all together back to this model, it may be that if increasing independence is one of the agents of change, of maturation in this system, children who experience parental neglect or parental deprivation may be exposed to a premature form of independence, which may be one of the reasons we're seeing this acceleration. And if so, then we wouldn't expect to see the same processes occurring during development. And so we would see this enduring phenotype, which would explain some of the larger group differences we see following exposures to early adversity, which might help explain some of the individual differences that emerge in emotion regulation behaviors that can exacerbate in adolescence. So to conclude, I've said that the neurobiology of human emotion regulation is very slow, but this slow pace can have its advantages and maybe tell us something about sensitive periods of development. Caregivers are a species expected stimulus for humans and may provide important social regulation during immaturity. And deprived caregiving can alter the neurobiology. It's one of the factors that can alter the neurobiology of emotion regulation. But perhaps we can better understand some of these changes by thinking about this as an accelerated pacing following stress and seeing the stress effects as developmental adaptations as opposed to simple aberrations. So with that, I would just like to thank members of the laboratory, people who've always been supportive of the work and the funding, but especially the families and individuals who have participated in these studies. And thank you for your time and attention.
For the stressed infants or the stressed children and the buffering effect, the individual variation you found in the last study you reported in terms of parental buffering, and to what extent are individual differences in that related to parenting behavior, parenting responsivity and sensitivity?
Yeah, it's such an important question, and we're obviously very interested in that question as well. We have, in that data set, not been able to identify what is different about those kids. So it may be something about the kids, too. There's a lot of heterogeneity in their backgrounds, and we haven't been able to identify anything yet. So I don't have a satisfying answer today. Those data are still being analyzed. And
have you used measures of attachment security in children that age? So you've looked at that, and you don't find any relationships?
Not in that
okay.
metric.
Okay. Mm-hmm. Can I ask a follow-up question?
Sure.
To what extent in children who are not coming from an abnormal environment, are you thinking about this as a potential biological mechanism to explain attachment security? Is that where you're headed with this?
It's a good question. So we haven't thought about it so frankly in attachment security the way that traditional attachment theory would talk about. But I think if we think about security more broadly as being one of the bases for emotion regulation, I don't think there's a tremendous amount of difference. So, you know, we are able to feel regulated when we feel safe. That's not the only factor, but that is certainly an important factor. So if we wanted to extend that to attachment security in a formal sense, then I think it's probably too early to say, but I think thinking about these connections between amygdala and prefrontal cortex might be a good candidate.
That was great. I'm thinking about all the different kinds of stimuli that you use as probes. So you've used fearful faces, you've used fear conditioning with the square and the noise and public speaking, and you're seeing similar patterns of response across different, so there's similarities in the stimuli at one level, but there's also differences. So I'm curious about, I don't know, your take on the generalization to these different classes, like subsets of these sort of fear-invoking kinds of stimuli. Mm-hmm. And then if you branch out of that group, what might you see if something's novel versus instantiating fear?
Yeah. So I think fear faces, I think really loud aversive stimulus, I think that being socially evaluated, I think that they're all really good probes of the system, but I don't, in this age group, in this population, I don't think it's specific to fear. So I think it's more generally about arousal involving social stimuli is actually what, well, it doesn't have to all be social, I suppose, but I think social stimuli are a really good way to activate the circuitry. So another behavior that I didn't talk about today that parents sometimes will report is indiscriminate friendliness. So this sort of over willingness to approach a stranger in children who experience caregiver disruptions. And so that too, I think, is another example. It's not a fear response, it's more like a difficulty regulating in the presence of this stimulus that's social. So I think it's broader than fear. It's just that fear happens to be a very good way to assess it.
You talked about, I really like this image of the amygdala knocking on the door of the medial prefrontal cortex. Mm-hmm. And I was wondering if you had any thoughts on other things that might drive that maturation of the medial prefrontal cortex.
Well, certainly puberty is going to be a big one. And that is, that's one area that we haven't gone into so much, in part because of the populations that we've been working with and the way that our questions have been shaped by the findings that we're getting. But we would expect that puberty might be involved. We haven't seen this in our data, but there are certainly findings out in the field that children who experience adversity may exhibit earlier pubertal maturation. And so if we're getting earlier connectivity here, could that be associated? Sure, but we haven't looked at that yet.
I just had a question about whether you've done studies or if you know of studies that have been done about like various parenting styles with kids, so not necessarily like adopted kids, but maybe kids with more authoritarian parents or where they actually have an adverse reaction to the parent being present.
Mm-hmm. Okay. So there is, everything that I talked about today was the parent buffering against this fear response. But there is another branch in our lab that we've just begun that is asking the other question of parents amplifying that response. So in the rat studies, the mother's anesthetized. In the human studies, the kids are wearing headphones, the parents are filling out questionnaires, so the parents aren't doing anything, right? So, so what happens if the parent is doing something? Or another way to ask that is your question, what about individual differences in parent-child relationships? So there's another great paper from Regina Sullivan's lab where she took the mom, rat, and she did the fear conditioning with the mom. So the peppermint odor was paired with mild foot shock. So the mom doesn't like the peppermint odor. Then she puts the mom into the nest with the pups, presents the peppermint odor, and the pups who didn't undergo fear conditioning, they learned to fear the peppermint odor. Why? Because the mom's behavior changed. She started acting anxious and overprotective of her pups. And in fact, at a time when amygdala would be non-reactive, when the mom is exhibiting that fear, they are prematurely showing this amygdala increase. So we are, the interpretation is not just that parents are buffering, but parents have a lot of power in regulating the circuitry, and I think that that's a really powerful message for thinking about how much parents can do for our kids. So now we're videotaping parents undergoing fear conditioning and showing that to kids in the scanner.
Can you comment at all on the role of the hippocampus, if any, in this particular form of learning?
Yep. So our lab has really not looked at the hippocampus all that much until recently, and it's becoming clear that we really can't not look at the hippocampus any longer. So in traditional rodent studies, the mantra is that in cued fear conditioning, you rely on the amygdala, and in context fear conditioning, you need the amygdala plus the hippocampus. In the human studies, the way that we're doing our conditioning, it's a discrimination learning. And so it's a little bit more complex. And what we've seen so far is in adulthood, we get a really nice coordination of amygdala, PFC, and hippocampus. In childhood, all you seem to need, based on the fMRI data, is the amygdala. And then you increasingly see the circuitry become more complex. In the post-institutionalized group, we are seeing the hippocampus coming online, participating in the learning at an earlier stage. And so we're trying to probe that also for its adaptive value.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.